Hello student, today I am going to teach you on the topic immunity to infection. Introduction. The term immunity is derived from the Latin word immunitas, which means exemption or freedom from taxes or public service. The applications of this term today in a medical context refers to ability of the human body to remain free of disease, which means the capability to resist infection of harmful microbes. The cells and molecules responsible for immunity constitute the immune system, and their collective and coordinated response to the introductions of foreign substances is called the immune response. The immune system comprises innate and adaptive immune responses coordinated to prevent infection. Innate immune responses are early responses based on preformed cells and effector molecules with a limited repertoire of antigen receptors. In contrast, adaptive immune responses take several days to mount but take advantage of a very large repertoire of antigen receptors and comprises memory. Types of immune system. Immune systems are broadly classified into two types. 1. Innate immunity or congenital immunity or non-specific immunity. 2. Adaptive immunity or acquired immunity or specific immunity. Both are not mutually exclusive. Innate immunity refers to non-specific defense mechanisms that come into play immediately or within hours of an antigen's appearance in the body. These mechanisms include physical barriers such as skin, chemicals in the blood, and immune system cells that attack foreign cells in the body. Adaptive immunity. Adaptive immunity, also known as acquired immunity, refers to antigen-specific immune response. The adaptive immune response is more complex than the innate immunity. The antigen first must be processed and recognized. Once an antigen has been recognized, the adaptive immune system creates an army of immune cells specifically designed to attack that antigen. Adaptive immunity also has a memory that makes future responses against a specific antigens more efficient. The adaptive immune response provides the vertebrate immune system with the ability to recognize and remember specific pathogens and to mount stronger attacks each time the pathogen is encountered. There are two classes of adaptive immune response: humoral immunity and cell-mediated immunity. In humoral or antibody-mediated immune response, B cells are activated to secrete antibodies, which are proteins called immunoglobulins. The antibodies circulate in the bloodstream and permeate the other body fluids, where they bind specifically to the foreign antigen that stimulated their production. Binding of antibody inactivate viruses and microbial toxins such as tetanus or diphtheria toxins by blocking their ability to bind to receptor on host cells. Antibody binding also marks invading pathogens for destruction, mainly by making it easier for phagocytic cells of the innate immune system to ingest them. In T-cell mediated immune responses, activated T cells react directly against a foreign antigen that is presented to them on the surface of a host cells or antigen presenting cell, also known as APC. Remarkably, T cells can detect microbes hiding inside host cells and either kill the infected cells or help the infected cells or other cells to eliminate the microbes. The T cells, for example, kill a virus infected host cells that has viral antigens on its surface, thereby eliminating the infected cells before the virus has had a chance to replicate. B cell activations. B cells are generated in the bone marrow, and it takes 1 to 2 weeks to develop from hematopoietic stem cells to mature B cells. The first exposure to a microbe or an antigen, either by infection or by vaccinations, leads to the activations of naive B lymphocytes. The immunoglobulin D or IgD and monomeric immunoglobulin M or IgM surface receptors of B cells bind to specific antigen and initiate the B cell activations. There are two types of B cell activations. One is T-independent B cell activations and second T-dependent B cell activations. T-independent B cell activation, many non-protein antigens such as polysaccharide and lipids stimulate B cells for antibody productions in the absence of helper T cells, and these antigens are called T-independent antigen or Ti antigen. Most Ti antigens are polyvalent, being composed of multiple identical epitopes. Such polyvalent antigens induce cross-linking of 2 or more surface Ig molecules on B cells, thereby deliver activating signals to the cells. Two membrane proteins, immunoglobulin alpha and immunoglobulin beta, that are linked by disulfide bonds to each other and covalently linked to membrane, transduces the signals generated by clustering of surface receptors. These two molecules, together with surface immunoglobulin, form the B lymphocyte antigen receptor complex. In addition, many polysaccharides activate complement by alternate pathway and generate C3d, which bind to the antigen and provide second signals for B cell activations. Responses to Ti antigens consist largely of IgM antibodies of low affinity and do not show significant heavy chain class switching, affinity maturation or memory. T-dependent B cell activation. Antibody responses to protein antigens require recognitions of antigen by the helper T cells or T cells and cooperation between the antigen-specific B cells and T lymphocytes. Antigen-specific B cells bind to native antigen through surface Ig receptors, internalized by receptor-mediated endocytosis, and process it in endosomal vesicles. The peptide fragment of the antigen is then presented along with MHC class II proteins on their surfaces, which is subsequently bound by T cell, resulted in the activations of T cells as well as B cells. As the internalized antigen is being processed, the B cell also expresses B7-1 and B7-2. Helper T cell that recognize MHC peptide complex on B cell binds to B7 molecules with its CD28 and gets stimulated to proliferate. Once activated by antigen recognition and co-stimulation, T cells expresses a surface molecule CD40L that binds to CD40 on the cell surface. This engagement results in initiations of enzyme cascades that leads to transcriptions of several genes, which subsequently induce activations of resting B cell. Following the activations of B cell, many Ig molecules highly specific for antigens are produced and B cells undergoes affinity maturations and some form memory. T cell activations. The precursors of T cells are produced in the bone marrow and mature in the thymus, so they are called T cells or T lymphocyte. T cells activations that leads to a cytotoxicity of target cells or stimulations of antibody productions by B cells needs two signals. Signal 1, interactions of an antigenic peptide presented on the MHC molecule with a TCR-CD3 complex. Signal 2, antigen non-specific co-stimulatory signal provided by interactions between CD28 on the T cell and B7 proteins on APC. T cells that express glycoprotein CD4 recognize antigen combined with a class II MHC molecule and generally function as helper T cells or T cells. T cells that express glycoprotein CD8 recognize antigen combined with a class I MHC molecule and generally function as cytotoxic T cells or TC cell. Moreover, MHC1 molecule is exclusively found in all nucleated cells and tissue, whereas MHC2 molecule is normally found in dendritic cells, macrophages, B cells, endothelial cells, and thymic epithelium. T cell activations. The central event in the generations of both humoral and cell-mediated immune response is the activation and clonal expansion of T cells. T cells activations is initiated by interactions of TCR-CD3 complex with a processed antigenic peptide bound to a class II MHC molecule on the surface of an antigen-presenting cells or APC. Interactions of a T cell with antigen initiates a cascade of biochemical events that induces the resting T cells to enter the cell cycle, proliferating and differentiating into memory cells or effector cells. T cell activations requires a co-stimulatory signals provided by APC. Interactions of B7 family members on APC with CD28 delivers the co-stimulatory signals. Engagements of the closely related CTLA4 molecule with B7 produce an inhibitory signal. B cells also serve as antigen-presenting cells in T cells activations. Resting B cells express class II MHC molecule but fail to express co-stimulatory B7 molecule. Consequently, resting B cells cannot activate naive T cells, although they can activate the effector and memory T cell populations. Further, antigen-presenting cells or APC stimulate T cells via interleukin-1 or IL-1, which also stimulate B cells growth, neutrophils, fibroblasts, and epithelial cells, while IL-6, which is also secreted by T cells, increases productions of immunoglobulin and increases productions of acute phase rectin mannose binding lectin, C-reactive protein, and fibrinogen. T cells stimulate APC by interferon gamma or INF gamma and also stimulate itself and other T cells by IL-2. Helper T cells could be separated into two classes depending upon the specific cytokines the cells secretes in response to antigenic stimulation. Th1 cells primarily produce interferon gamma and interleukin-2, whereas Th2 cells produce IL-4, IL-5, IL-6, IL-10, and IL-13. TC cell activations. Microbes can infect and survive in a wide variety of cells. The functions of killing cells with microbes like virus in the cytosol is mediated by CD8+ cytotoxic T lymphocyte, which is also known as CTLs. In addition to their role in defense against microbes, the second important functions of CD8+ CTLs is the eradication of many tumors. The activations of naive CD8+ T cells require antigen recognitions and other stimulatory signals and proceeds in steps like those for other T cells responses. There are three major signals for TC activations. First, loose attachments of non-specific interaction between LFA1 integrin of T cells with ICAM1 of APC. Second, recognitions or firm attachments of T cells receptor with antigen present on class I MHC molecule. Third, activations by Th1 cells via interleukin-2. It also recognizes non-self class 1 MHC during non-genotypically identical human transplants due to polymorphism in structure between individuals, which resulted in the acute rejections of organ allografts. When exposed to infected or dysfunctional somatic cells, TC cells release the cytotoxins perforin, granzymes, and granulysins. TC cells causes cytolysis of target cell by the actions of pore-forming protein tubule called perforin inserted in the cell membrane. Granzymes enter the cytoplasms of the target cells through perforin, and their serine protease function triggers the caspase cascade, which is a series of cysteine proteases that eventually lead to apoptosis or programmed cell death. A second way to induce apoptosis is via cell surface interactions between the TC and the infected cells. When a TC is activated, it starts to express the surface protein FAS ligand or FAS-L, which bind to FAS or APO1 or CD95 molecules expressed on the target cells and thereby induce apoptosis of the target cells. In conclusion, many disease-causing pathogens keep infecting our body, but owing to immune system, we resist and survive without noticeable symptoms. All these protections are attributed to a special white blood cells, which is also known as lymphocyte, present in our body. Basically, lymphocytes are of two types, B lymphocyte and T lymphocyte. They play a pivotal role in destroying and eliminating foreign pathogens like bacteria, viruses, etc. B lymphocytes are responsible for antibody productions and humoral immune system. T lymphocytes stimulates B lymphocyte and other white blood cells, including T lymphocyte itself, and trigger immune response, which is called cell-mediated immune system. If any of these immune systems stop working, we will be susceptible to many unwanted diseases, which thus pose many threats to our survival.